Hello again everyone, and this is Cloning and Genetic Engineering Part 2, in which we look at Part 2 of the title, that is Genetic Engineering. So what is genetic engineering? Well, it's not cloning. It's also known as genetic manipulation or genetic modification, or often simply GM these days, and it's different from cloning because cloning just replicates genes that already exist. You are copying another organism. But GM can produce a completely new set of genes, and they don't have to be even in the same species. With cloning, genes must stay within the same species, but genetic modification can move genes between species. In fact, one of the ways that we do it is by copying genetic information from other species into bacteria. So we have actually been practicing genetic engineering for a long time, thousands of years in fact, and one way of genetically engineering animals is selective breeding. You can also selectively breed plants, I suppose. That's what Mendel did. So when we choose the best animals to breed from, we produce young that have the same traits as those animals, and we can experiment with this and breed one good animal with lots of different animals and hope that we get great animals when genes come together in a good way. But nowadays, genetic engineering, in the true sense of the word engineering, uses enzymes to literally cut up and rewrite sections of genetic code. So you literally cut the chromosome at a certain point and insert a section of DNA. So, how do we do this? Well, roughly speaking, we remove the cell nucleus and we use enzymes to cut out those genes that we want to keep. And then we also remove a section of DNA from a bacteria, and we replace it with the section of code that we took from our DNA. And we replace that into the bacteria cell, and we now have a modified bacteria. And then it can now reproduce normally, and it will copy the section of DNA that we want. So we can reproduce that section of code many, many times. So how can we use GM? Well, it should be obvious, but every type of organism can be changed in any type of way. Now, literally that means we can do anything to any creature, which kind of sounds horrible. So we need to learn more about genes to understand how they work, so that we know how to successfully change genes and change them in a way that is responsible. So, as you can see, this has also got some quite big questions going along with it, but we've already grown GM vegetables that are more resistant to disease and also grow faster, and we have started to experiment with transferring traits between species of animal and not just in plants. So one thing that we've tried to do with genetic engineering is gene therapy, and we've tried this on humans. Gene therapy involves inserting copies of a normal allele into chromosomes of someone who carries a faulty allele. So you find the gene that causes whatever disorder they have, and in a healthy cell you cut out the normal allele. You make many copies of that allele by inserting it into bacteria, as we saw before, and we insert the healthy allele into the cell of the person who has the disorder. Now, that is to change one cell. So we have to decide exactly how we're going to do this, and how we try to do gene therapy is to absorb the material through fat-based nasal sprays into the nose. Sometimes we directly inject genetic material which may be accepted and replicate inside one of the patient's cells, or we can modify cold viruses which infect other cells with their DNA, and if we put the target allele into this cold virus, then we can have our virus infect target cells with the new fixed allele. So unfortunately, gene therapy is not a reliable technology. The techniques we use can get the alleles into the body, but there is very little to guarantee that the alleles, when they get into the patient's cells, will join to the DNA or will join to them in a way which allows them to work properly. So also, gene therapy doesn't rewrite more than a few cells, and so it's likely that the treated cells will be crowded out through the process of cell division by the person's natural cells, which are untreated and carry the genetic defect. So before we finish, we just need to talk about the Human Genome Project, because this has really been the driving force in genetic research for many years. It was the collection and organization of the entire human genome. It was begun last century, and the collection of the data took yet 10 years, and they are still working on the analysis of the data today. So the goals of the project were to decode and order all 3 billion base pairs on the 23 human chromosomes. It was also to identify all the different genes that are present in the genome, and also to develop faster methods for sequencing DNA. That was probably to help with their research. So nowadays we are trying to understand exactly what the order of the base pairs means, and we have decoded some, but not all of them. There are 3 billion of them after all. DNA fingerprinting is a useful technique that comes from being able to sequence DNA. It was started in 1985, and it's a forensic technique which helps police tell where people were and if an object belongs to someone, or at least has contacted them. So if you take a person's DNA, you will find that it is completely unique to every other person's DNA. So it's like a fingerprint, hence the name. So that about wraps us up. We've looked at cloning and we've looked at genetic engineering, and we've scratched the surface of both. We haven't looked at some of the more advanced techniques in genetics, and we haven't looked at any of the most recent developments. So if you're interested in genetics, there's an awful lot more to learn.